Imagine playing a really high-stakes game of hide and seek. But in this specific version of the game, the hider is a deadly disease.
Right. And the hiding spot they've chosen inside your body makes them completely invincible to our absolute best medical weapon.
Exactly. I mean, you know they're in the house. You have the tools to defeat them, but you physically cannot reach them, which is just the frustrating, heartbreaking reality of treating acute myeloid leukemia.
Yeah, it really is.
But we're looking at some fascinating new research today that suggests we've been, fighting this disease from totally the wrong angle.
Which is a pretty bold claim, right?
Yeah.
Considering we spent, the last 50 years in oncology basically trying to build bigger, better bombs.
Right, like stronger chemos.
Exactly. We develop incredibly powerful chemotherapies and these targeted biologic agents. But the harsh clinical reality of acute myeloid leukemia, or AML, is that you can literally nuke the body with chemo, get a patient to what looks like total remission.
And then it just comes back.
Yeah. Months or years later, the disease just comes roaring back, and it is entirely because of these microscopic hideouts.
Okay, let's unpack this. Because to understand how to trap this cancer, we first have to map the geography of its favorite hideout, right?
We do.
So, the source we are diving into today completely maps out where these cells are hiding deep inside your bones. And what I want you, the listener, to take away from this deep dive is how scientists decoded the molecular GPS these cancer cells use.
And crucially, how we can actually smash that GPS to force the cancer out into the open.
Yes. So let's start with the culprit itself, because we aren't just talking about any cancer cell here.
No, we were talking about leukemic stem cells.
LSCs.
Okay, LSCs. Break that down for us.
Sure. So in any tumor or blood cancer, you have the bulk tumor cells. Think of those as the regular foot soldiers.
Yeah.
But then you have the generals, right? The stem cells.
The ones pulling the strings. Exactly. These LSCs are responsible for initiating the disease, driving its progression, and ultimately causing those devastating relapses. Think of them as the roots of an incredibly invasive weed.
Oh, I like that. So standard chemotherapy is like mowing the lawn.
Exactly like mowing the lawn. Yeah. You can mow it all you want, wiping out millions of the bulk cancer cells above ground. But if you leave the LSC roots intact beneath the soil, well, the weed just grows back.
Often stronger.
Oh, almost always stronger and more resistant.
And the reason chemotherapy can't kill those roots ties into a biological concept called quiescence, right?
Yes, quiescence.
Let's make sure that mechanism is crystal clear for everyone listening, because standard chemotherapy is essentially, it's a poison that targets actively dividing cells.
That's the fundamental mechanism. Yeah, it targets the cell cycle.
Right. So it looks for the cellular machinery that pulls DNA apart to make a new cell, and it destroys it.
But LSCs have evolved this terrifying defense mechanism against that. They simply go to sleep.
Which is the quiescence.
Exactly. Quiescence is a state of deep cellular dormancy. They stop dividing entirely. They lower their metabolism to an absolute crawl.
So because they aren't actively copying their DNA?
The chemotherapy drugs just essentially wash right over them. The drugs literally don't recognize them as a threat. They are invisible.
Wow. Okay, so the researchers here needed to find out exactly where these cells go to sleep. They had to look inside the bone marrow.
Which is not what most people think it is.
No. I mean, I always used to picture bone marrow as just a big hollow tube filled with this homogeneous red biological soup.
A lot of people do. But it's actually highly structured. It's a really complex, multi-tiered ecosystem.
So how do we visualize it?
Well, let's use a bit of visual memory. Picture a long bone, like your femur. Okay? The bone marrow inside is divided into two key longitudinal zones.
Okay, got my femur pictured. What's zone 1?
First, you have the metaphysis.
Yeah.
This is the spongy trabecular bone near the rounded ends of the bone.
The ends. Got it.
Right. And the researchers further divided this into proximal and distal regions. Basically, the top end and the bottom end. That's the metaphysis.
And zone 2?
Zone 2 is the central marrow, which is exactly what it sounds like. It's the vast open cavity right in the middle of the bone shaft.
Okay, so the researchers wanted to see where the cancer stem cells were physically hanging out within that specific geography.
Yes, but finding a few sleeping stem cells in a sea of billions of normal, healthy, blood-forming cells.
Sounds impossible.
It's like finding a specific grain of sand on a massive beach. They couldn't just look through a microscope and spot them. They had to track them using a molecular barcode.
Oh, this part is so cool. Let's explain this barcode. Because you can't just rely on how a cell looks, right?
No, in cellular biology, looks are deceiving. You have to read the proteins that are sticking out of the cell's surface.
So they scanned for a highly specific combination of these surface proteins.
Exactly. By scanning this barcode, they isolated the most dangerous malignant leukemic stem cells in the entire body. In the paper, they refer to these as LGMPs.
LGMPs, what exactly does that stand for?
It stands for leukemic granulocyte macrophage progenitor-like cells.
That's a mouthful.
It is. But the barcode is what matters. To be classified as an LGMP, the cell had to hit a very specific checklist. It had to be GFP positive, but Lin negative.
Lin negative meaning it doesn't have the markers of a mature cell.
Right. And it also had to be SCA1 negative, but positive for c-KIT, CD34, and a specific receptor called Fc gamma R23.
Okay, so a ridiculously specific barcode.
Ridiculously specific, but it worked perfectly.
And when they tracked these highly aggressive leukemic cells in mice, using that barcode, the visual was just stark.
Completely. These dormant cells were not floating randomly in the soup. They were heavily concentrated at the extreme ends of the bone.
Metaphysis?
Yeah, the proximal and distal metaphysis. They were actively avoiding the wide-open central marrow.
Why there? Did they cluster there on purpose?
Absolutely on purpose. Look, when the researchers ran an experiment where they extracted leukemic stem cells from the metaphysis and then extracted a different batch from the central marrow and transplanted those distinct groups into healthy mice.
Let me guess. Different results.
The results were night and day. The cells taken from the metaphysis caused a drastically more aggressive, lethal leukemia.
Man. Let's lock this in with an analogy for everyone listening. Picture the metaphysis, those spongy ends of the bone, as an exclusive, highly fortified luxury bunker.
A luxury bunker. I like that.
The cancer stem cells navigate into this bunker, they lock the heavy steel doors behind them, and they go into deep hibernation.
And by sleeping in this bunker, they're essentially hooked up to a life support system. It maintains their stemness, keeping them lethal and ready to strike years later.
While the central marrow, meanwhile, is just a chaotic open battlefield. It's totally unprotected. The cancer wants no part of it.
Which completely reframes the clinical approach. The bunker isn't just a location. It's an active participant in the disease.
Yeah.
It shields the cancer.
Right. Okay, so let's just do a quick recap for you listening to make sure we have this foundation set. Basically, AML stem cells, the roots of the cancer, preferentially hide out in the extreme ends of your bones to stay dormant, protected from chemo, and highly malignant.
Spot on.
Which brings up a massive question for you listening. Think about this. If you were an oncologist looking at this map, how would you even begin to coax these sleeping cells out of their bunkers so your chemotherapy could actually hit them?
I mean, you can't just yell into the bunker. You have to figure out how they found the bunker in the first place.
Right. What is the molecular compass guiding them there?
And that takes us to the mechanism. This is where the biology just gets unbelievably elegant.
The molecular tug of war. Let's get into it. Because the researchers identified a localized three-part system that dictates exactly where these cells anchor. Let's break down the three players. Who is player one?
Player one is a protein called CXCL12. This is a chemokine. Its entire job is to create a chemical scent trail.
So CXCL12 is the magnet, or the scent.
Yeah, think of it as a powerful magnetic beacon. It diffuses through the tissue, and cells that have the right receptor will follow that gradient from a lower concentration to a higher concentration, right to the source.
So the bone marrow naturally uses this.
It does. It naturally secretes this to organize healthy blood cells. But the leukemia cells hijack it. They are magnetically drawn to it.
Okay, so player one is the CXCL12 magnet. What's player two?
Player two is DPP4.
DPP4.
Right. This is an enzyme, specifically a peptidase, that is actually expressed on the surface of the AML cancer cells themselves. And DPP4 acts like a pair of hyperspecific molecular scissors.
Okay, wait. A peptidase. Explain the scissors part. How specific are we talking?
Very specific. So proteins are long chains of amino acids, right? And their 3D shape dictates their function. It's a lock and key mechanism.
Okay.
The DPP4 scissors specifically snip off just 2 tiny amino acids from the very tip of the CXCL12 magnet, the N-terminus.
Just 2 amino acids.
Just 2. But that tiny cut changes the shape of the magnet just enough that it no longer fits into the cancer cell's receptor.
Oh, wow.
By making that cut, the beacon is completely inactivated. The scent trail just goes totally cold.
Okay, wait. I need to push back here because that makes zero sense to me.
Why?
Think about it. The environment is putting out a magnet to attract the cancer cell, but the cancer cell is walking around covered in scissors that destroy the magnet.
Yep.
If the cancer destroys the very beacon guiding it, how does it ever find the bunker? Wouldn't it just wander aimlessly?
That is the exact paradox the researchers had to solve.
Yeah.
And the answer is player 3, GPC3, or Glypican-3.
GPC3, okay, what does that do?
This is an endogenous shield. It physically binds to the DPP4 scissors and blocks them from making their cut.
Okay, so we have the magnet, the scissors, and the shield. How does this actually play out structurally inside the bone? Who is holding what?
Inside that metaphysis bunker, there's a very specific type of support cell. It's an N-cadherin positive mesenchymal stromal cell.
Okay, let's just call them the bunker architects for short.
Bunker architects works. These architects secrete both the CXCL12 magnet and the GPC3 shield.
Oh! Here's where it gets really interesting. The hyperlocal geography. So the architect cells release the beacon to attract the cancer cell.
Right.
The cancer cell arrives, carrying its scissors, completely ready to chop up the beacon and move on. But before it can, the architect provides the shield, jamming the scissors.
Exactly. And because the scissors are jammed, a very dense, perfectly intact pool of the magnet builds up. But only in the microscopic space right next to that architect cell.
Only right next to it.
Yes, if the cancer cell drifts even a millimeter away into the central marrow, there is no shield.
Ah, so its scissors immediately activate.
Exactly. They activate, destroy the local magnet, and the cell is completely adrift until it floats back into a shielded zone.
It's a biological tether.
It is.
The bone marrow provides the magnet and the shield, the cancer brings the scissors, and together they create this microenvironment perfectly engineered to trap the cancer cell in a state of suspended animation.
And this explains a century of oncology struggles.
Yeah.
We were looking at the cancer cell under a microscope in total isolation.
Just staring at the bad guy.
Right. But a cancer cell is not an island. It relies entirely on this localized tug of war with its immediate neighborhood to survive.
So just to do a quick recap of this part, the stromal cells and the ends of the bone use a chemical magnet to lure the cancer in and a biological shield to stop the cancer's own enzymes from destroying that magnet.
It's a perfect trap.
It really is. So let me pose a thought exercise to you, the listener. Knowing this incredibly delicate three-part balance, what do you think happens to the entire system if we, say, reach in and permanently snap off the cancer's scissors?
Yeah. What if we take away the DPP4? That is the exact question that drove the next phase of the research.
And what did they find?
By genetically deleting the DPP4 enzyme in these leukemic mice, the scientists triggered a massive system-wide cascade. It altered the entire physical distribution of the cancer across 3 distinct scales.
Let's walk through those scales, because the chaos caused by removing one microscopic pair of scissors is just mind-blowing. Start with the largest scale, the whole body. What happens systemically?
At the systemic level, we look at the peripheral blood and other organs. Normally, as leukemia progresses, the cancer cells overpopulate the bone marrow and spill out into the bloodstream. Right. They invade the spleen and the liver.
And that organ failure is often what kills the patient.
Exactly. But when they deleted the scissors,
What happened?
without the scissors constantly chewing up the magnet, the magnetic gradient inside the bone marrow wasn't being managed at all. It spiked. The pull inside the bones became so overwhelmingly strong that the gradient between the bone marrow and the blood actually reversed.
Wait, reverse?
Yes. The AML cells became physically trapped inside the bones. They couldn't escape into the bloodstream at all. The spleens of these mice were completely spared, and their healthy blood counts, like hemoglobin and platelets, remained perfectly intact.
Wow. So by removing the scissors, the luxury bunker turns into a maximum security prison.
That's a great way to put it. But it gets worse for the cancer because of what happens at the second scale, inside the bone marrow cavity itself.
Right, because inside the bone marrow, that delicate balance between the spongy metaphysis and the hollow central marrow is destroyed.
Completely. Because the magnet was now everywhere, not just shielded in the bunkers, the cancer cells lost their specific compass. They were flushed out of the metaphysis and washed right into the chaotic, unprotected central marrow.
And the third scale, down at the actual cellular level.
Because the hyperlocal signaling was disrupted, the leukemic stem cells physically let go. They detached from the architect cells. They completely lost their physical lifelines.
Okay, so let's summarize this. We've trapped them inside the bone, we've flushed them out of their bunkers, and we've stripped them of their support system. But why is that actually lethal? What happens to a sleeping leukemic stem cell when it's left floating in the open central marrow?
Well, without the suppressive, calming signals of the bunker, the cancer cells wake up. They lose quiescence.
They wake up.
Yeah. The researchers tracked their metabolism and found hyperactive cell cycling. The cells were suddenly dividing at a frantic pace.
Okay, I look at this as the exhaustion trap. Imagine taking someone who is fast asleep in bed, you wake them up, and instantly force them to run an ultramarathon at a dead sprint.
Good luck with that.
Right. But you give them no water, no shade, and no calories. They burn out fast. By pushing these cancer stem cells out of their sleep state, they are forced to divide rapidly without the local resources to sustain that division.
The metabolic math just doesn't work for them at all. And to prove this, the researchers ran something called serial replating assays.
Let's define that. What's a serial replating assay?
It's a classic laboratory test. You take stem cells, you let them form a colony in a dish, then you extract them and force them to form a new colony again and again and again. It basically tests their stemness, their endurance.
And how did the cells without scissors do in this test?
They failed miserably. They completely lost their capacity for self-renewal. Because they were forced to sprint continuously, they triggered their own internal self-destruct mechanisms.
Apoptosis.
Yes. Massive rates of apoptosis, programmed cell death. They literally exhausted themselves to death.
Man, so just by deleting the DPP4 scissors, the cancer is trapped, it's flushed into the open, forced to divide, and burns itself out. It's a brilliant biological domino effect.
It really is.
But as a consumer of science, I have to play devil's advocate here. Because cancer is notoriously unstable, right? It mutates rapidly.
Sure.
So if you just take away its scissors, how do we know the cancer won't just mutate a new way to survive? How can we definitively prove that the bone marrow neighborhood is the master controller here and not just some internal rewiring of the cancer cell itself?
Look, that is exactly the kind of pushback rigorous science demands. You can't just tweak the cancer and call it a day. You have to prove it from the other side of the equation.
How do you do that?
You have to leave the cancer completely untouched, and instead you mutate the neighborhood.
Ah, a mirror image experiment.
Precisely. The scientists engineered a highly sophisticated conditional knockout mouse model.
Conditional meaning what?
Conditional meaning they don't delete a gene everywhere. To spare you the deep genetics jargon, they used a system to selectively delete the gene that produces the CXCL12 magnet.
Okay.
But here's the kicker. They didn't delete it from the whole mouse. They deleted it only inside those specific architect cells in the metaphysis.
Okay, wait, so the rest of the body is totally normal, but the bunker suddenly loses its beacon.
Yes. Then they took completely normal, wild-type, highly aggressive leukemia cells with their scissors fully intact and transplanted them into these newly mutated mice.
Okay, the suspense is killing me. What happened? Did the cancer find a way around it since its scissors were working?
No. The exact same exhaustion trap occurred.
Wow.
Without the local beacon from the architect cells, the cancer couldn't anchor in the metaphysis. So the cancer cells drifted out into the central marrow, they woke up, cycled rapidly, and exhausted themselves to death.
So it's conclusive.
This is the definitive biological proof. The environmental gradient is the absolute puppet master of the leukemic stem cell's fate. The cancer is a slave to its geography.
That is just staggering. It proves the tumor is really only as powerful as the soil it's planted in.
Exactly.
But I know what everyone listening is wondering right now. Because mouse models are great, but translating a genetic knockout in a mouse to a human patient usually takes, what, decades of clinical trials?
Usually, yes.
So are we just looking at a cool science trick that won't help humans until 2045?
And this is where the clinical stakes of this paper become monumental.
Yeah.
We don't have to wait decades to figure out how to disable the DPP4 scissors in humans.
Why not?
FDA-approved DPP4 inhibitors already exist.
Wait, really? For what?
Type 2 diabetes.
Are you serious?
Dead serious. Drugs like sitagliptin are used in clinics globally every single day to manage blood sugar by inhibiting this exact same enzyme.
Wow.
The safety profiles are already established. The drug is literally sitting on pharmacy shelves right now.
So you're saying we could take a drug used for diabetes, give it to a leukemia patient, and theoretically snip the cancer's scissors, flushing it out of the bunker?
That is the exact clinical horizon this paper is pointing toward. Oncology is moving heavily toward these mobilization strategies.
Meaning moving the cancer around.
Exactly.
Yeah.
What if we use a DPP4 inhibitor to jam the scissors? Or we use a drug called AMD 3100, which is a CXCR4 antagonist, to basically blindfold the cancer cell so it can't sense the magnet at all.
You just rip them right out of the bunker.
You combine these niche-disrupting drugs with standard chemotherapy. You force the cancer out into the open central marrow. You wake it up from its quiescent state, so it's actively dividing, and then you hit it with the chemotherapy.
The same chemotherapy that targets dividing cells.
Exactly. Suddenly, the chemo that used to wash right over the sleeping stem cells becomes devastatingly effective.
Because now they are awake, their metabolism is redlining, they are highly vulnerable to toxins, and they have absolutely nowhere to hide. I mean, this isn't just an incremental step, it's completely reframing the battlefield. Which leads me to ask you, the listener, a question. If we can medically control where a cancer cell is allowed to live inside the body, how does that fundamentally rewrite the rules of treating chronic diseases?
It's huge. It forces us to stop viewing cancer as an isolated invader and start viewing it as an ecological problem.
Yeah. So what does this all mean? Let's bring everything we've unpacked today full circle. We started by mapping the geography of the bone marrow, discovering that the ends of our bones, the metaphysis, act as luxury bunkers, while the central marrow is a chaotic, open battlefield.
Right.
We zoomed in to uncover the invisible, hyperlocal tug-of-war between the CXCL12 magnetic beacon, the DPP4 molecular scissors, and the GPC3 shield.
Such a delicate balance.
And finally, we saw the definitive proof that if you disrupt this delicate ecosystem, either by jamming the scissors or removing the magnet, you initiate a forced eviction that leads directly to cancer stem cell exhaustion and death.
And if we connect this highly specific cellular mechanism to the absolute biggest picture possible in medicine, it leaves us with a truly provocative reality.
What's that?
For over a half-century, the entire global field of cancer therapy has been obsessed with finding the perfect poison. The ultimate, highly toxic agent designed to kill the cancer cell directly.
Right.
But this research proves that a cancer cell is truly only as deadly as the neighborhood that harbors it.
It's molecular real estate. Location is everything.
It really is. If we can manipulate the geography of the human body, if we can act as biological real estate agents and permanently evict cancer from its luxury bunkers using drugs that already exist, could we turn the body's own architectural layout into the ultimate weapon against relapse?
And it makes you wonder, what other dormant diseases in other organs are just quietly sleeping in their own specialized bunkers, just waiting to be evicted?
It's a fascinating thought.
It is, and that is something I want you, the listener, to mull over today. Because the next time you think about a stubborn disease hiding in the body, remember that they aren't invincible supervillains. They are just playing a highly complex game of hide and seek. And thanks to this kind of rigorous cellular mapping, we are finally bringing the sledgehammers to tear down their hiding spots. Thanks for joining us on this deep dive.